step of cell respiration is a process that occurs in the cytoplasm called glycolysis. Glycolysis is a pathway in which glucose is divided and converted into pyruvate, which is the ionized form of pyruvate acid. To be more specific, glucose, a 6-carbon sugar, is divided into two 3-carbon sugars. These smaller 3-carbon sugars are then oxidized and the remaining atoms are arranged to form 2 molecules of pyruvate. Glycolysis is divided into 2 phases: energy investment and energy payoff. During the energy investment phase, the cell actually uses 2 ATP. However, this energy investment is repaid for the production of 4 ATP during the energy payoff phase. The energy payoff phase works by using a process called substrate-level phosphorylation. During this process, ATP is formed through the enzyme-assisted addition of phosphate groups to ADP molecules. During glucose oxidation, NAD+ acts as an electron carrier to temporarily store energy. This reduction reaction causes NAD+ to be converted into NADH. The total net energy yield from glycolysis per glucose molecule ends up being 2 ATP plus 2 NADH. All of the carbon from the glucose molecule are accounted for in the 2 molecules of pyruvate, so no CO2 ends up releasing through glycolysis. Glycolysis does not actually require oxygen to work, although if oxygen is present, the energy that is stored in the pyruvate and the NADH can be carried and extracted through the citric acid cycle and oxidative phosphorylation.
In this video, I'll be talking about pyruvate oxidation. When glucose enters the cell, it first goes through glycolysis in the cytoplasm, breaking down into 2 molecules of pyruvate, also known as pyruvic acid. This process generates a small amount of ATP and produces NADH. The pyruvate then moves into the mitochondrion where it undergoes pyruvate oxidation, which requires oxygen. Each pyruvate loses a carbon atom, releasing carbon dioxide. The remaining 2-carbon piece attaches to a coenzyme A, forming isotyl-CoA, which enters the citric acid cycle, also known as the Krebs cycle. In this cycle, acetyl-CoA is further broken down, producing more NADH and a small amount of ATP. The NADH created during these processes carries high-energy electrons to the electron transport chain, where a significant amount of ATP is produced. Overall, this series of reactions connects the breakdown of glycolysis to energy production in the presence of oxygen.
The Krebs cycle, also known as the citric acid cycle, plays a key role in energy production. It takes place in the mitochondria, where the cycle begins when a molecule called acetyl-CoA combines with another molecule called oxaloacetate. Together, they form a molecule called citrate or citric acid. This step is highly exergonic. Next, the citrate is rearranged into a molecule called isocitrate through a simple structural shift. Third, a redox reaction occurs, where the isocitrate is oxidized. In doing this, the positive NAD is reduced. This releases carbon dioxide and forms ketoglutarate. After this, another redox reaction occurs, resulting in the molecule being oxidized by reducing that positive NAD and is made reactive by the addition of CoA. This reaction forms succinyl-CoA. Next, the succinyl-CoA forms ATP by inserting GDP and exerting GTP. This step removes the CoA, which leaves the molecule as just simple succinate. Now, the succinate is oxidized, and the FAD is reduced by being inserted into the molecule and exerted as FADH squared. This results in a molecule called fumarate. Following the redox reduction, H2O is added into the molecule, which forms a malate molecule. Lastly, the malate molecule is oxidized by the reduction of NAD+. This leaves the molecule as the original oxaloacetate, where the cycle starts over again. The cycle excretes carbon dioxide, which we exhale. It is an interesting process which happens continuously to keep you alive.
Here's a quick overview of the final steps of cellular respiration known as oxidative phosphorylation. This process includes the pathway of electron transport, the process of energy gradient driving cellular work known as chemiosmosis, and the production of ATP. Together, these processes produce the 26 to 28 ATP molecules. Here's a closer look at the pathway of electron transport. At the beginning of the chain, the NADH2 contributes 2 electrons. As the electrons travel through the chain of multi-protein complexes, the electrons follow a pattern of reduced and oxidized states as they accept or donate their electrons. The electrons move in a downhill direction through the proteins, and in this diagram, the second large protein contains FADH2. The FADH2 contributes its own 2 electrons as well. At the end of the chain, 2 electrons finish the process and reduce molecular oxygen. This aids in the production of H2O from 2 proteins and half of an oxygen. This brings us to the ATP synthase process known as chemiosmosis. A definition for chemiosmosis is the energy stored in the form of a hydrogen ion gradient across a membrane used to drive cellular work, such as the synthesis of ATP. In this process, the hydrogen ions in a high concentration outside of the membrane flow into the synthase complex. They rotate once within the membrane, then enter the mitochondrial mix through a channel structure. The rotation of the synthesis complex activates catalytic sites in the lower portion of the structure, which in turn converts ADP and P into ATP. This is all due to the hydrogen gradient known as proton motive force. This concludes the process of oxidative phosphorylation.
For this video, I'll be drawing alcohol fermentation. Alcohol fermentation occurs when glucose is broken down in the absence of oxygen. It starts with glycosis in the cytoplasm, where 1 glucose molecule is converted into 2 molecules of pyruvate, generating a small amount of ATP and NADH. Since oxygen isn't present, the pyruvate is transformed into ethanol and carbon dioxide. During this process, NADH is oxidized back into NADH+, allowing glycosis to continue and to produce more ATP. This type of fermentation is common in yeast and some bacteria. Unlike alcohol fermentation, lactic acid fermentation also begins with glycosis but converts pyruvate into lactic acid instead of ethanol, which occurs in certain bacteria and muscle cells when oxygen is limited. Overall, both fermentation processes enables cells to produce energy without oxygen, but they produce different end products, which I will draw on the right side of the screen right now.